As I understand it, as soon as the genome of the COVID-19 coronavirus was published, you were ready, at least in terms of a digital model, with the vaccine within days. You had the solution, I mean, sitting there on a computer screen, and it's like all you needed to do was manufacture it, distribute it, get it, a regulatory approval. Was that frustrating to know that you essentially had the answer, but it was locked in a digital version, and now the obstacles were all physical?
I would say in a sea of frustration we all felt during the pandemic, I'm not sure that incremental frustration could be attributed to that only because we were all overwhelmed trying to find solutions. Instead, it gave us a purpose, which was as rapidly as possible, take that sequence, make it, which we did in days physically, test it in animals, which we began to do within a week. And then based on data from animals, we ended up being ready to go into humans within some 40 days. And in March with the NIH, we began human trials of this vaccine. That time frame, albeit slower than the 1 or 2 days for the digital rendering, if I can call it that, of the ultimate vaccine. Nevertheless, that time frame was so abbreviated that we felt we were accomplishing something, and it was leveraging this platform we had spent $2 billion in the prior 10 years developing. Thereafter, we had to test it in humans in much larger trials. And I would say, so you understand the mindset in the biotechnology pharmaceutical industry, we did not have the vaccine until the results from our phase 3 trial were there. Because you need to show statistically that compared to a placebo control, you are having not only the efficacy, but also the safety needed for it to make sense to be widely deployed. And I would say only at that moment did we know that 10 months earlier we had the sequence. Because until then, we may have had the sequence for nothing. That discipline exists in biotechnology development very, very rigorously. Last year, when I kept hearing about people worrying about cutting corners and all that, I mean, it's so counter to how everybody that I know in this industry behaves, because we are dealing with people's lives, and it is absolutely incumbent on us, first and foremost, to make sure we convince ourselves that this is something that's safe and efficacious. The regulators help us do that. The clinical community helps us do that, but it is in our best interest. Otherwise, the company's future is completely affected.
I mean, there's a couple of things that you said there that strike me as pretty extraordinary. Number one, over 10 years, you spent $2 billion with no idea how it was going to work out. That's a lot of money on what feels like something of a moonshot. During those 10 years, were you certain that investment was going to pay back, or is that just part of the calculus of Flagship Pioneering?
It's a good question, but I want to make sure that I'm clear about what we did and didn't know. When you build a platform, and this is to me among the underappreciated parts of what the advancement in our field looks like. I mean, in tech, platforms are well understood and they're highly enabling. In biotech, platforms are almost non-existent. The people think of them as technologies you use in the laboratory, PCR, CRISPR, this and that. But what we mean by platform is a common set of technologies, understanding that together allows us to generate product after product after product in a very reliable, rapid fashion. That platform is what we developed over a long period of time. But the key there is that you don't develop the platform to have one product. So what we do to mitigate the unknown, if you will, is to have a multitude of products that are simultaneously advancing in clinical trials, so that the ones that actually make it through the clinical testing gauntlet, if I can call it that, are the ones that ultimately reinforce the platform and make it valuable. And so when you say, were you uncertain about what would come out, I would say we were not uncertain about the possibility that some of what we were working on would actually have impact. We just couldn't tell you which one, because of the vagaries of the human physiology and the fact that we don't have a good computational model yet of that. So it's important to understand that if you develop a technology that has one shot on goal, in our industry, something that I really don't like very much, there's this notion of shots on goal. It's believed that so few things work that you have to just take a lot of shots. We don't view it that way. We think that platforms endow you with multitude of chances. You pursue them all, as many of them as you can afford, and that's the maximum. That's the best way to maximize an outcome. We couldn't tell you it would have been for coronavirus. We couldn't tell you if it's going to be our next product in CMV or in heart disease, but we're working on all of them.
The second thing that you said that relates to what you just described that I found fascinating is that by March, you had human trials going. Now, that's March 2020, correct?
Yes.
Now, March is when most of us found out that we were in the midst of a pandemic. March was the month everything changed for the whole world, and you were already in human trial. Now, from that point forward, most of us understood that there might be some vaccines. There was sort of this Cheshire cat grin going on with Dr. Fauci. You know, it seemed like something was up, and there were various, promises that things might come within a certain time frame, depending on which politician you listen to. The time frame was further out, or which scientist you listen to. But you were behind the scenes. Take us behind those scenes into that 9-month sprint or 6-month sprint to get to where you could confidently say, we have a vaccine that works and we're going to be distributing it, and here's the plan. What happened in that time in between in 2020?
Well, keep in mind that most of the economy was shut down, and our team at Moderna was working feverishly, literally 24 hours a day in shifts, to be able to ramp up manufacturing. Keep in mind also that prior to the beginning of last year, we had largely deployed mRNA vaccines in maybe some 2,000 human subjects, and here we were ramping up to be able to make literally hundreds of millions of doses in the first year of operation. And so that is a no small feat, probably even a more giant feat than some of the scientific things we were able to do, because it was without any precedent to go from zero to that kind of scale and capability. So that was going on behind the scenes, in front of the scenes. I mean, this was a mad kind of dash to do that. It's remarkable that the manufacturing process stood the stress test, not only in our US manufacturing facility, but also we transplanted it to Switzerland through a partner and set up a capability there. So all of those things were preoccupying a large part of a heroic group of people. At the same time, we needed to design clinical trials. We needed to test some of the kind of key questions about a vaccine and its potential harmful effects so that we could rule them out. Very, very specific experiments we wanted to do to assure ourselves that we weren't taking extraordinary risks. So all of that was going on. But you're right in capturing the moment that, it's not that we had confidence that this would work. We just couldn't figure out why it wouldn't work, except everybody told us that it wouldn't work. And of course, history suggested it wouldn't work. And worse yet, history suggested that things that have never been tried before work even less somehow, apparently. And so what we needed to do is, frankly, block some of that out and just do the best science we could do. We didn't need to win the war of prediction. We just needed to win the war of demonstration. And if we were not successful, frankly, we were hopeful that other people would be successful so we could benefit from their work. And indeed, it was the case that the two mRNA vaccines that were advanced, basically using the exact same technology, ultimately did reach the sufficient efficacy and safety goals. But along the way, rightly, the scientific community was highly skeptical that this could be done, or done quickly, or done definitively, or done safely. And the burden was on us to do all of those. And if we were standing at the end, then we had the next race to enter, which was scale, distribution, access around the world, and then variants, which we could see coming already in the early days, and how do we prepare for those?